Okay, so welcome to this next video in which we are discussing the vascular endothelial growth factor receptor. Okay, so we're currently in the process of looking at the pathways downstream of the vascular endothelial growth factor receptor 2. Okay, so we've discussed that what's going to happen is this phosphatidylanosyl-3 kinase enzyme, which has this special SH2 domain, which is capable of binding to phosphotyrosine residues, is going to bind to phosphotyrosine residues on the cytoplasmic domain of the vascular endothelial growth factor receptor 2. And then is going to be perfectly positioned underneath the inner leaflet of the phospholipid bilayer to act on its substrate, which is within the phospholipid bilayer, which is these PIP2 molecules, and it will convert them from phosphatidylanosyl-4,5 bisphosphate PIP2 into phosphatidylanosyl-3,4,5 trisphosphate PIP3. Okay. And these PIP3 molecules are then going to carry the signal on. Now, before we go any further, I should just mention that PIP3 molecules are not a normal component of the phospholipid bilayer. And this is because there is an enzyme whose sole function, or one of its functions, I don't know if it's its sole function, but one of its functions is to break down phosphatidylanosyl-3,4,5 bisphosphate molecules, which are within the membrane, and return them to phosphatidylanosyl-4,5 bisphosphate molecules. Okay, so PI3 kinase takes them this way. There is another enzyme which is constitutively expressed that takes them the opposite way. And this enzyme is usually abbreviated to PTEN, and this stands for phosphatase antensin homolog. So where shall I write that? Okay, I'm going to go up here to write it. So PTEN stands for, the P is for phosphatase, okay, and then it's phosphatase and tensin homolog. So that 10 is for tensin basically, and tensin homolog. Okay, so that's the origin of the name PTEN, and basically this enzyme works on phosphatidylanosyl-3,4,5 trisphosphate molecules which are in the lipid bilayer and converts them back into phosphatidylanosyl-4,5 bisphosphate molecules, so that you don't usually have any PIP3 molecules within the lipid bilayer of the cell membrane. Okay, so when the phosphatidylanosyl-3 kinase enzyme becomes temporarily activated, then it will now it will convert phosphatidylanosyl-4,5 bisphosphate into phosphatidylanosyl-3,4,5 trisphosphate. So you'll get a temporary burst of PIP3 molecules within the inner leaflet of the phospholipid bilayer, and these are now going to carry the signal further. So let's see what's going to happen. Basically, certain enzymes are going to bind to these PIP3 molecules in the lipid bilayer of the cell membrane. Okay, so let's draw another PIP3 molecule out. So here are the two long-chain carboxylic acids. Here's a phosphate group, the inositol molecule. Okay, and then coming off the inositol ring, you then have these three phosphate groups, like so. Okay, right, so let's add some colour onto it because it makes it look simpler if we've got colour. Okay, so here are the long-chain carboxylic acids. Here is the glycerol molecule, this green horizontal line. The phosphate groups are all in vivid purple. Okay, like so. And then the inositol molecule is in blue here. Okay, right. Now, basically, certain enzymes have a domain which is capable of binding to PIP3 molecules. So just as the Sark homology 2 domain was capable of binding to phosphotyrosine residues, there is a conserved domain which is capable of binding to PIP3 molecules, okay? And this conserved domain which is capable of binding to PIP3 molecules, which I'm showing here, is known as the plextrin homology domain. So in pink here, this is a plextrin homology domain. so often abbreviated just to a pH domain. This stands for plextrin homology domain. Okay, so again, plextrin is some protein which contains a domain very similar to this, which is capable of binding to PIP3 molecules. Okay, and the name of this enzyme that is coming and binding to the PIP3 molecules that are now in the inner leaflet of the phospholipid bilayer is known as phosphoinositide-dependent kinase 1. Okay, so this is phosphoinositide, and then it's dependent kinase 1. And phosphoinositide-dependent kinase 1 is often abbreviated to PDK1 for short. So P for phosphoinositide, D for dependent, K for kinase, and then it's PDK1. Okay, now, not only does the binding of the plextrin homology domain to the PIP3 molecules that are within the lipid bilayer of the cell membrane sequester the phosphoinositide-dependent kinase 1 enzyme at the inner leaflet of the phospholipid bilayer, but it also actually activates its kinase activity. And once activated, it is a serine-threonine kinase enzyme. So we're going to see a lot of serine-threonine kinase enzymes looking at these pathways. So I think I might as well spend some time discussing what serine-threonine kinases do. So let me just show you the structures of serine amino acids and threonine amino acids. So we'll start off by drawing the structure of a normal amino acid residue. So this is the core structure of an amino acid residue. And then the R group of a serine amino acid residue contains a methylene group, then with an alcohol group coming off it. Okay, so this is the structure of a serine amino acid residue. And then also we'll have a threonine amino acid residue here. So here's the amino group again, the alpha carbon, the carboxylic acid group linked to the amino group of the amino acid, the next amino acid along, and then the R group is going to be similar to serine, but basically we're going to take one of these hydrogens off and replace it with a methyl group. Okay, so here's a methyl group, you have an alcohol group coming off, and then a hydrogen also coming off. And this is the structure of a threonine amino acid. Okay, and you'll notice that both of these have alcohol groups coming off, and just like the alcohol group of the tyrosine amino acid can be phosphorylated, these two amino acid residues can be phosphorylated to create phosphoserine residues and phosphothreonine residues. Okay, so that's what I mean by saying that phosphoinositide-dependent kinase 1 is a serine-threonine kinase. I mean it's going to stick phosphate groups onto the alcohol groups of the serine and the threonine amino acids via phosphoester links. Okay, right, we now have an active phosphoinositide-dependent kinase. Now, what's its target going to be? Well, basically, it's going to target another enzyme that is also going to get sequestered at the phospholipid bilayer by binding to a PIP3 molecule. So let me now draw another PIP3 molecule here. Okay, so again, here are the long-chain carboxylic acids, the glycerol molecule, the phosphate group, the inositol ring, and then the three phosphate groups coming off the inositol ring, and again we'll colour the structure in. So we have our long-chain carboxylic acids in orange here, we have our glycerol molecule in green, we have our phosphate groups in pink, all four of them, and then we finally have our inositol ring in blue. Okay, and basically another enzyme is going to have a plextrin homology domain, which is going to bind to these this other PIP3 molecule here, because of course you don't just produce one of them, you produce many of them. Okay, so on another PIP3 molecule you have got another enzyme here. Okay, and this enzyme again has a plextrin homology domain, which I'll colour in pink here. Okay, and Basically, this next enzyme is a very famous enzyme. This is the AKT enzyme, and AKT is its old name. It's the name that, if you read the primary research literature, they use for it. But its more modern name is to call this protein kinase B, and I would say that protein kinase B is taking over as its more famous name. Okay, so protein kinase B, and for short, of course, protein kinase B is usually denoted as PKB. Right, so protein kinase B also has one of these special domains known as the plextrin homology domain, which is capable of binding to a PIP3 molecule. Okay, and this means that when you produce this PIP3 in the inner leaflet of the phospholipid bilayer, that the AKT or protein kinase B enzyme is going to bind to this and get sequestered at the inner leaflet of the phospholipid bilayer. Now, it's perfectly positioned thus to be the target of this phosphoinositide-dependent kinase 1 enzyme. However, you might think, well, surely, why does it need to be phosphorylated? If it's come up here, it surely will be activated too now by the binding of the plextrin homology domain to the PIP3 molecule. But that's where AKT is different to phosphoinositide-dependent kinase 1. Phosphoinositide-dependent kinase 1 comes and binds to the PIP3 molecule, gets sequestered at this special location, and then also becomes activated by the binding of the plextrin homology domain to the PIP3 molecule. Whereas AKT just gets sequestered, it doesn't get turned on by the PIP3 molecule. To get turned on, to get activated, it needs to have a serine residue phosphorylated by the phosphoinositide-dependent kinase 1 enzyme. So it will have a phosphate group added onto a serine residue here. Okay, so here's a phosphate group now added onto AKT, and now you have an active protein kinase B enzyme. Now, so far we have reduced this entire pathway down to the activation of protein kinase B. That's where it's taken us. All of these details were all about activating protein kinase B. So now, what is the activation of protein kinase B going to achieve? Well, basically, it's going to activate a complex known as mTORC1. Okay, and I'm going to discuss this complex with you before I actually discuss how AKT is going to actually activate it, because this is quite a complicated complex. Okay, so mTORC1 stands for, this is a long name, the little m is for mammalian, the capital T is for target, the O is for of, the R is for rapamycin, so the mammalian target of rapamycin, and then the C is for complex, and then of course 1 is just for 1. So we are discussing the mammalian target of rapamycin complex 1. Now this is not just one protein, this is not just the mammalian target of rapamycin. It does include the mammalian target of rapamycin, but it has a bunch of other proteins attached onto it as well. So, let's now have a look at the proteins that are in this mammalian target of rapamycin complex 1. So the first one is the mammalian target of rapamycin, mTOR, okay, which is a protein. Now, you also have some other proteins in there, so I'm not going to write out that one's name because, you know, we've seen it here. It's the mammalian target of rapamycin. So you also have some other proteins attached onto the mammalian target of rapamycin. Okay, and the first one I'll mention is a protein known as RAPTOR. So RAPTOR stands for the regulatory associated protein of mTOR. So I'll write this over here. So the R is for the regulatory. The A is for associated. Okay. And I've just realised I'm about to write into where I want to put another protein later, so I better skedaddle over here. And then it's the regulatory associated protein, so the P is for protein, and then the TOR is for of mTOR, so of the mammalian target of rapamycin. So that's what Raptor stands for, which is quite a cool name for a protein. Then we have another protein over here which has a less cool name. Okay, and this is MLST8, which stands for mammalian lethal with Sec13 protein 8. Okay, so they do have quite ridiculous names. Regulatory associated protein of mTOR was the most sensibly named one. Okay, so MLST8 stands for the mammalian lethal. So the M is for the mammalian, the L is for lethal, and then it's Sec13. So the S is for Sec13, and then this is protein. So where they get the T from, whether they just take the T from protein, I don't know, but it is abbreviated to MLST8. And then it's the mammalian lethal with and Sec13 protein 8. And to make this look simpler, I'm going to color code it. So we'll have the mammalian lethal and Sec13 protein 8. We'll have that underlined in red and that outlined in red over here. Okay, we'll have the mammalian target of rapamycin, the protein after which the entire complex is named, labelled in green here. Okay, and we'll have, what other nice colour do I have? Orange. We'll have the regulatory associated protein of mTOR in orange here. Now, this is not the mammalian target of rapamycin complex 1 finished yet. We have 2 more proteins to go. Okay, so the next one is a protein known as DEPTOR, which means domain containing mTOR interacting protein. So this is DEPTOR. Okay, so this stands apparently for domain containing mTOR interacting protein. So the D is for domain, then it goes off a bit because we've got containing mTOR, mammalian target of rapamycin in full, interacting protein. Okay, so how they quite got DEPTOR from that, I don't know, but that's supposedly what DEPTOR stands for, the domain containing mTOR interacting protein. Okay, so we'll have that one labelled in blue here. So I'll underline its name in blue and I'll outline it in blue. And then the final protein, well actually I should just say this is the functional complex. There is one final protein that I'm going to add on to this, but really this is the functional mammalian target of rapamycin complex 1. Often you'll see people just, write this as the mammalian target of rapamycin complex 1. However, in the inactive mammalian target of rapamycin complex 1, you do have one further protein stuck on, but this one's going to be removed when we actually activate this complex. Okay, so you have one more protein here, which is PRAS40. Okay, right, so what should I outline PRAS40 in turquoise, I think. Right, so basically, the activation of protein kinase B is going to lead to the activation of this mammalian target of rapamycin complex 1. Okay, but this isn't really understood that well. What is known is that it phosphorylates PRAS40. Okay, and when you phosphorylate PRAS40, PRAS40 falls off the mTOR, basically. It falls off the complex. Okay, so you go back down to that that foursome, basically, Raptor, mTOR, DEPTOR, and MLST8. You lose PRAS40. Okay, so you go to the active complex, but that alone is nowhere near enough to actually activate the mammalian target of rapamycin complex 1. So yes, it's part of the activation, but it's not enough alone, basically. So PRAS40 does fall off, and it is due to phosphorylation by AKT, but that's not really the main mechanism by which AKT activates this. So I'm going to tell you what is known about the mechanism by which AKT activates it, but there will be some holes, basically. Okay, so in order to discuss how protein kinase B, or AKT, is going to activate this, we need another player here. We need something known as the tuberous sclerosis complex. Okay, so let's turn our attention to studying the tuberous sclerosis complex. Okay, so the tuberous sclerosis complex of proteins consists of two members. So there are two members of this elite little club, and they are the tuberous sclerosis complex protein 1, the TSC1, and the tuberous sclerosis complex 2 protein, the TSC2 protein. Okay, and when together, these two proteins in this complex catalyze a certain reaction. And the reaction they catalyze is the turning off of a certain G protein, a certain monomeric G protein. Okay, so there is a certain monomeric G protein known as the Reb G protein. And like all G proteins, Reb has two states. It has an on state where it is bound to guanosine triphosphate, GTP, and it has an off state where it is instead bound to guanosine diphosphate. And basically what the tuberous sclerosis complex does to the Reb G protein is that it turns it from the on state where it has GTP bound to it, and it turns it instead into the off state, basically. So it catalyzes the conversion of Reb GTP, the on Reb, into the off Reb. So it turns Reb off. Okay, so that's what this complex does. Now, basically, what protein kinase B is going to do is it's going to phosphorylate this tuberous sclerosis complex protein 2. Okay, so here is tuberous sclerosis complex protein 2, and it gets a phosphate group stuck on it, and when tuberous sclerosis complex protein 2 gets a phosphate group stuck onto it, there is another protein that now loves to come and bind to the phosphorylated tuberous sclerosis complex 2. Okay, so along comes some other protein here, which comes and binds to this tuberous sclerosis complex 2. And this protein has a rather bizarre name. It's called 14-free-free. Okay, so this protein that I will highlight in red to make it a little bit more distinguishable than its poor name makes it, is the 14-free-free protein. Okay, so 14-free-free will bind to the phosphorylated tuberous sclerosis complex protein 2. And when you have 14-free-free bound to the phosphorylated tuberous sclerosis complex protein 2, it can no longer bind to the tuberous sclerosis complex protein 1. So the tuberous sclerosis complex, hence, is no longer made. So basically what protein kinase B is going to do is it's going to stop these tuberous sclerosis complexes from being formed by phosphorylating the tuberous sclerosis complex 2 protein, which will then force it to associate with this 14-free protein. Now, if there are no functional tuberous sclerosis complexes, then the Reb G protein no longer gets turned off. So what you get is an accumulation of Reb GTP within the cytoplasm because there's no enzyme complex to turn it back off. So you get an accumulation of on Reb G protein within the cytoplasm, and then this accumulated Reb G protein in the on state then somehow activates the rest of the mammalian target of rapamycin complex, and this is the stage that isn't known, that isn't understood at all. Basically, we don't know how Reb GTP accumulating within the cytoplasm turns the mammalian target of rapamycin complex 1 on. Okay. But then the overall effect then now is that we have this mammalian target of rapamycin complex 1 turned on, okay? And we will come back to discuss what this does right at the end, but basically it is a transcription factor. And the reason we're not going to talk about it now, and we're going to turn our attention to other pathways downstream of the vascular endothelial growth factor receptor 2, is that all of the other pathways are also going to lead to the activation of transcription factors. So what instead we'll do is look at all the other pathways and then combine them all together at the end and say, look, we've got a whole collection of transcription factors, we can now summarize all of their effects as so. So we'll come back to the active mammalian target of rapamycin complex 1 later. Okay, in the next video we'll turn our attention to a different pathway that is downstream of the vascular endothelial growth factor receptor 2.